So in the last couple of videos, we've really looked at taking our time, looking at all of the organelles, again, from the inside of the cell all the way to the outside of the cell, working our way outward. So now we finally made it to the plasma membrane. We aren't going to talk about the structure of the lipids in the plasma membrane just yet, but instead we're going to talk about the extracellular matrix. So here we can get a pretty good image of the complexity of the extracellular matrix, but let's break it down and start getting to know some of the components. So the extracellular matrix, which we are specifically talking about now in animal cells, has many functions. This includes maintaining the structure of the cell. It also includes kind of giving the tissues different identities and keeping them distinct, as well as intercellular communication. So while the specific composition of the extracellular matrix can vary widely between different cell types, there are a lot of things that we have in common between cells. One of these is collagen. So you may have heard of collagen in reference to things like skin elasticity. This is a structural protein that we'll talk about in the next slide. Fibronectin is also something that we find in most extracellular matrices around cells. Integrins, which are inherently embedded in the plasma membrane. You can see that they have connections on the outer side of the cell and the inner side of the cell, which will come in handy. And proteoglycans and other carbohydrates. So proteoglycans, which we'll talk about in more depth in a bit, have both a carbohydrate and a protein component. We also have polysaccharides as well. So let's start by going in depth for each of these components. So again, collagen is a protein that provides structural support. These collagen fibers connect to the rest of the extracellular matrix through a protein called fibronectin. So you can see collagen, it's very rope-like, connected through fibronectin to integrins and other membrane-bound proteins that are going to thread through the plasma membrane. So you can see this just forms just a connection here. I think about it like nectin, like connecting this up. Okay. Integrins can also do a lot of other things. So they're not just there to hold the fibronectin. They can also facilitate cell signaling and the adhesion of cells to surfaces in other cells. So let's look at next the proteoglycans. So you can see just from this image that they're very complex, but let's get in there and see what they're made of. So again, proteoglycans, as the name suggests, if you think of the proteo part as being for proteins, and glycans, this is going to be polysaccharides, kind of a carbohydrate, protein-carbohydrate combined molecule. So in our proteoglycan, we have a polysaccharide chain in the middle. So this is the green part. And from that, you have branches of protein, which are going to be more lavender. You can see the core protein here, and then smaller carbohydrates branching off of it. So it's a highly complex branch structure that contains largely carbohydrates, but also proteins branching off of it. We can also have other polysaccharides that are not proteoglycans. And a notable one is hyaluronic acid, which you may hear about in reference to skincare. So a lot of these extracellular matrix type proteins we hear about a lot when we think about things that are used for anti-aging purposes. So the wide variety of proteins and proteoglycans found in the extracellular matrix allows different cell types to actually have extracellular matrices with varying degrees of rigidity. So for example, the extracellular matrix that you find in bone or tendon types of tissue might be different than something like the brain, which is softer. So that contributes as well to the actual structure of the surface and at the organism level, the tissue sort of structure as well. There's also some interesting evidence that the extracellular matrix has a role in tissue repair and regeneration. So again, thinking about sort of these ideas that, again, structural proteins, polysaccharides like hyaluronic acid, we see a lot of sort of this repair sort of motif, even like repairing like wounds and things. And also this can be involved in tissue differentiation. So becoming one type of tissue as versus another type of tissue. So you can see that we have a lot of different components here that can really help give us characteristic cell types as well as help with cellular signaling. So we're not going to talk too much about any of these extra things in depth, but we can also trap signaling molecules within the extracellular matrix to help the cells respond to signals more efficiently. And we can even control the level of hydration based off of some of the ultimately osmotic properties of things like proteoglycans. So it can actually indirectly attract molecules or ions that will attract water and keep things hydrated. So there are a lot of different uses for the extracellular matrix. We're not spending nearly as much time as we probably should on the extracellular matrix, but for now we should just know that it's an incredibly important part of the cell. It's around all animal cells, and we're able to provide structure, support, and a lot of other functions for the cells. With all of this going on at the outer surface of the cell, it may be difficult to get communication going between different cells in the vicinity. So in order to combat this challenge, we have a lot of intracellular junctions in animal tissues. So there are three main types of intracellular junctions in animal cells: tight junctions, gap junctions, and desmosomes. So here we're going to be looking at a few different types in different cells. And again, while these can all be present in one cell at the same time, different cell types may have different proportions depending on what that cell's function might be. So let's look at tight junctions first. So tight junctions, as the name suggests, form a really tight seal between cells. So what we're actually looking at here is going to be kind of the junction between two cells, and you can see they're almost like sewn together with these proteins. So it's composed of proteins. The main purpose of this is to prevent fluids from leaking into the space between cells. So you can see how tightly these cells are woven together. If you would think about liquid kind of being up here in the space, you wouldn't want it to leak between cells. So this is useful in many situations. So things like intestinal epithelia, for example. You don't want what's running through your intestines to leak between cells. Okay, you can again think of this as the cells being stitched together by many proteins. Our second type of intracellular junction is a gap junction. So gap junctions form tiny protein-lined channels between cells. They're sort of actually a not-so-secret passage if you're a molecule. So you can see the gap junction. Here it's cut in half. This is just kind of a hollow tube through which things can diffuse. So this is not going to be any kind of like active or necessarily selective transport. This is more going to allow diffusion between cells. So things that couldn't otherwise pass through the membrane, things like ions, which we'll talk about in Chapter 7, larger things, even like proteins, might be able to diffuse through, nutrients included. So this can really help kind of communicate between adjacent cells. I think ions are a really important use for this. So our third type of intracellular junction is desmosomes. So desmosomes, as you can see, are very intense. So we've got this sort of almost kind of like spider-like structure sticking out into the cytoplasmic space, kind of riveting these cells together. It's just as terrifying to behold when we look at the microscopic image. So desmosomes are actually composed of intermediate filaments, which we've just kind of talked about in a previous video, and we know now that these are most often used in situations where we really want to create sort of a permanent structure. We don't want to be assembling it and disassembling it. So I think the use of intermediate filaments in a desmosome is really appropriate. And the times when we'd want to use this are when the cells need to withstand some type of mechanical force. So you can think of things like muscle contractions. When all of the muscle cells are connected together and they're all trying to contract, they need to stay attached to one another. Another example is stretching, maybe something like in your skin. So you'd want to make sure that those cells don't come apart when there's a lot of movement or stretching. So things that undergo a lot of mechanical stress. And it's possible for cells to, again, have more than one type of junction between neighboring cells. Plant cells have a different situation. They don't have the kind of extracellular matrix that we would think of for animal cells. They instead have the protection of a cell wall that will be composed of structural carbohydrates like cellulose and hemicellulose, probably also some things like lignin that we won't get into now. So you can see the structure of the cell wall. Our primary cell wall, which is usually quite a bit more narrow, thinner sort of cell wall, and secondary cell wall, which forms underneath, and the cell can just deposit it as it goes over time. So depending on the type of plant, it can continue to just keep depositing secondary cell wall. And of course, the thicker the secondary cell wall, the more like woody type of plant you'll have. We can also glue this together with a gel-like substance called the middle lamella, which is going to consist of things like pectin, which if you've made jam during the quarantine, you may know that pectin can be used kind of to like really gel up things. So the middle lamella is again going to be kind of a gel that will sort of adhere everything together. So the cell wall does a lot of things besides just protect the cells. It helps the plants resist turgor pressure when they're going to be taking in water or when they're drying out a little bit, like the plants in my office, and other mechanical forces. So plant cells, just like animal cells, need a way to talk to each other, but it's a little bit more challenging with the cell wall in place. Okay, again, we do have a plasma membrane, but it's buried underneath all of these structural carbohydrates. So what they have instead is a channel that goes through the cell wall. It's lined with plasma membrane, so it's almost like continuous cells just sort of attached together. These channels are called plasmodesmata. And plasmodesmata can allow access to things that diffuse from one cell to the other. You can actually see this between cell types. So again, here's our plasma membrane. It's the interior of one cell. You can even see the cell walls here. And these plasmodesmata, again, they're not protein lined like a gap junction. They're membrane lined, but they do have a similar function. You can even have transcription factors and other types of things like that go through these plasmodesmata. So this is actually very useful to the plant. So we do have channels and we do have some outer protection. So in conclusion, there are three main types of junctions in animal cells and one main type in plant cells. So these would be, again, the gap junction, the tight junction, and the desmosome in our animal cells, and then plasmodesmata in our plant cells. These junctions can keep cells watertight, allow for diffusion between cells, and hold cells together. And again, cells can have all three types. They may have them in different proportions depending on its function. For example, skin cells must maintain a watertight barrier and probably need a tight junction, but they also have to resist stretching forces, so they may have some desmosomes as well.